Hello, everyone. This is Jiang Liu from GemPharmatech, and we are welcome to today's webinar. It is a special day today, Rare Disease Day. Rare diseases, as opposed to common diseases, refer to a category of illness with relatively low prevalence. According to the statistics, there are currently over 7,000 rare diseases types recognized in different countries and regions, posing a significant threat to human health. By utilizing gene editing techniques, GemPharmatech has independently developed and validated a series of mouse models for genetic and rare diseases. These models have greatly contributed to scientific research and drug development. And here are the rare diseases models we have, including thalassemia, fiber diseases, hemophilia, and so on. And we also provide the mouse models such as KO, gene humanized mice. We have established over 20,000 strains of KO mice, which also makes us possess the largest collection of genetically engineered mouse models. We also have mouse models replicating human diseases like cancer, metabolic diseases, neurodegenerative diseases. We also offer services, including custom model generation, preclinical services, and customer breeding. For preclinical services, we have 6 platforms for therapeutic areas like cancer, metabolic disease, and neural diseases. If there is any need, please contact us with no hesitation and accelerate your innovation with GemPharmatech. And let's begin our webinar, our speech part now. After the speech, we will have a panel discussion with our R&D scientist, Dr. Binzhou Han. Today, we will talk about biological functions of migrasomes with our guest speaker, Dr. Dong Jiang. Migrasomes are newly discovered organelles which are generated by migrating cells. The formation of migrasomes has been observed in various in vivo settings, and the migrasomes have been shown play important roles in organogenesis, coagulation, and mitochondrial quality control. And before the speech, I will do a brief introduction of our guest speaker. Dr. Dong Jiang obtained his PhD degree at Tsinghua University, whose mentor is Professor Li Yu, and became a postdoc fellow since then at Tsinghua University. His research focuses on the biology in migrasomes, newly discovered organelles which are generated by migrating cells, particularly on the physiological and pathological functions of migrasomes in embryonic development, tumor metastasis, immune response, coagulation, and other processes, as well as their diagnosis and treatment potential. He has published several high-impact research articles in NCB, which was the cover story, and the Cell paper for Twice, and so on. And Dr. Jiang is also our client. He uses a hemophilia model F8 KO mouse, which is a rare disease model to investigate biological function of migrosome. Now let's welcome Dr. Jiang.
Yeah, okay. Yeah, okay. Thanks for Jiamin for your invitation and introduction. And I'm Dong Jiang from Tsinghua University. And I'm very glad to be here to share our research with you. And as the Jiamin's introduction my research is focused on the physiological and pathological functions of migrasomes. And today I'm going to talk about the biological functions of migrasomes, particularly in the development and the GemPharmatech and the coagulation context. So, firstly, I will introduce what is migrasomes. So 10 years ago, our lab discovered a new cellular structure. As you see here, that during the cell migration, cells will leave long protrusions from the cell, we named with the retraction fiber. And then many smaller vesicles will emerge from the tips and the junctions of the retraction fibers. So considering the cell migration dependence and the smaller vesicles-like structure, we named these vesicles with migrasomes. So if we come back to the history of the discovery of migrasomes, we initially observed the debris-like structures outside of the cells under the electron microscopy. And I believe that most of the investigators who performed the electron microscopy has observed these vesicle-like structures. But in most cases, nobody cares about this observation. So, but if when we improve the quality of the samples and get a high resolution image, as you can see here that we find these structures, these vesicle structures that have a very clear morphological characteristic that numerous smaller vesicles are wrapped in a large vesicle which is connected to a retraction fiber. So when we observe these structures, we go back to the literature to find whether anyone has reported or published some similar structures, but we can't find it. So we started to realize that maybe these structures, especially the vesicular structure we named with migrasome, maybe newly and never have been reported by anyone before. So and we also can observe this vesicle and the retraction fiber-like structures under the scanning electron microscopy, which is consistent with those observed under the transmission electron microscopy. So when we realize that maybe this is newly and never been reported before, we're trying to isolate these structures using the gradient certification. And when we get this sample, we performed the mass spec analysis, and we try to find out the proteins enriched in these structures. When we get the mass spec data, we try to identify the protein marker of the migrasomes. And we finally find a protein which is a tetraspinin 4, which is highly enriched in the migrasomes. As you can see in this picture, this beautiful picture, that this is come from a fibroblast cell, which is expressed in with tetraspinin 4 GFP. So you can see that tetraspinin 4 is enriched in the migrasomes and can label the migrasomes in very beautiful manner. So when we observed these structures, so many scientific questions emerged from our observation, such as a fundamental question at that time is whether migrasomes are existing in living organisms and whether migrasomes perform functions in the physiological process. So to answer these questions, we urgently need to find an animal model to study migrasomes and uncover the physiological roles of migrasomes in the physiological context. So, considering to the cell migration dependent of the migrasomes, we know that cell migration is a very critical element in most of the physiological or pathological process, such as the embryonic development and the tumor metastasis or the immune response, especially in the embryonic development and the gastrulation stage, all of the cells migrate, as you can see the first video. that all of the cells migrate, and the cell movement leads to the formation of endoderm, mesoderm, and the ectoderm. And the cells migrate to a definite location and interact with the surrounding cells and as well as the circumstance to generate a microenvironment and modulate the morphogenesis. So based on the imaging systems and the cell migration context, we finally chose and tested the migrasome observation in the zebrafish embryo, especially in the gastrulation stage because the cell migration is featured. So we just synthesize the tetraspanin 4, which is a migrasome marker protein. We synthesize the tetraspanin 4 mRNA in vitro and inject this mRNA into the embryo at the 8-cell stage and to label the migrasomes. And then we put the embryos under the confocal microscopy to observe the formation of migrasomes. As you can see that we find a lot of migrasomes generated by the embryonic cells during the gastrulation stage. And we find that after the migrasomes generated by the cells, they will detach from the retraction fiber to be a free-floating migrasomes. And if we take a 3D image using the spinning disk microscopy, we can get a more striking structure of migrasomes and retraction fibers. And then we have mentioned that migrasomes will detach from the retraction fiber to be a free-floating migrasomes. And we find they will accumulate to some specific location and within the embryo, just like that migrasomes will accumulate in some cavity in the embryo to form some migrasome-enriched center. So this observation indicates that maybe migrasomes can provide some spatial information. So when we get these observations and we demonstrate that migrasomes exist in living organisms, that in the zebrafish embryo development. So the next question is, what's the functions of migrasomes in the development? So to investigate the roles of migrasomes in the embryo development, we need to delve into the mechanisms of migrasome formation and to identify the regulating genes of migrasomes. So we need to identify the regulated genes and generate the knockout animal and to see what will happen. So fortunately, the other lab members has identified tetraspanins, such as tetraspanin 4 and tetraspanin 7 is a key regulator genes for migrasome formation. So based on this discovery, we generate the tetraspanin 4 and tetraspanin 7 knockout zebrafish. And then we check the migrasome formation in this mutant. So we can see that in the Y-type embryos, there are many migrasomes are generated by the embryo cells and distributed in the extracellular space. But in the tetraspanin 4 or tetraspanin 7 knockout embryos, the migrasome formation has reduced significantly. So a big question is what will happen to this tetraspanin mutant? So the final phenotype is a very long story, and I wouldn't like to speak too much here. But then what we got is randomized organ morphogenesis defect. Especially most of the defect is coming from the left-right axis establishment. And of course, we believe that just because we didn't check enough, that probably have a lot of more defect in this tetraspanin mutant. So in this case, for example, in the Y-type embryos, almost all of the fish shows a single liver, as we shown here that we label the liver with C. And but in the tetraspanin mutant zebrafish embryos, some fish present duplicated livers, just like this one and like this one. And in another example that the, in the Y-type embryos, they should have two kidneys, But the kidneys were labeled with the SLC20A1A, but in the terraspinning knockout zebrafish embryos, some fish presents just have single one kidney, and most of the fish present a morphogenesis defect in the kidney development. And in some fish, they should they should they should have the left-right symmetric distribution of the organ pattern, but in some fish, they present the organ pattern reverse, just like this mutant fish. So when we get these phenotypes, a big question is emerging that we can't rule out the possibility that these phenotypes are caused by tetraspanids in migrasome-independent manner. So we really worry about how could we see tetraspanic phenotype is due to migrasomes and not just because this gene, because we know that every gene in the living organisms play multiple roles in different contexts. So we need to demonstrate that this phenotype is due to migrasomes. So eventually we designed a rescue experiment that beginning we don't believe it will work. Yeah, we just purified the migrasomes from the wild-type embryos, and then we inject these purified migrasomes into the tetraspanic knockout embryos before the gastrulation stage, and then we stained the embryos with the organ markers and check the organ morphogenesis. And we find that most of the defect of morphogenesis defects in tetraspanic knockout embryos can be rescued by migrasome injection. This data indicates that the phenotype, which is the morphogenesis defect phenotype, is due to the migrasomes. And I will explain why this rescue experiment can work. So if we summarize this phenotype in the tetraspanic knockout embryos, we can see that the left-right symmetry defect, such as the organ pattern reverse and the organ duplicate. And we also can see that the morphology defect of organs, such as the smaller kidneys and the elongation organs. So in this case, we focused on one particular phenotype, which is left-right laterality defect in the axis formation. And we need to explain how this phenotype is raised and regulated by migrasomes. And from the literature, we know that in the zebrafish embryo development, they will form a temporary organ named the Kupfer's vesicle, which contains a lot of cilium. And this cilium will swing along the clockwise flow and distribute the left-right signal to the proper location. So if the Kupfer's vesicle formation is defect, so the left-right symmetry will occur some defect phenotype. And we also, regarding to the Kupfer's vesicle, we know that Kupfer's vesicle are formed by a group of cells which is named with dorsal forerunner cells, or DFCs. The DFCs keep a tight cluster, and move along with the embryo axis. So the first thing, of course, the first thing we checked is the Kupfer's vesicle phenotype in tetraspanic mutant. And you can see the Kupfer's vesicle is seriously defect in tetraspanin 4 and tetraspanin 7 mutant. In the Y-type embryos, the Kupfer's vesicle is normal, but in tetraspanin 4, tetraspanin knockout embryos, the Kupfer's vesicle becomes very smaller, and the number of ciliums reduces significantly. So, which presents the Kupfer's vesicle formation defect in this mutant. And then we checked the dorsal forerunner cells migration, which can explain why the Kupfer's vesicle formation defect. So we established the transplantation assay that we just transplant the Y-type dorsal forerunner cells from the Y-type embryo to another Y-type or tetraspanic knockout embryos and to check the migration pattern. And we find that in the Y-type embryos, the DFCs keep in a tight cluster and move along with the axis. But in tetraspanian 4 or tetraspanian knockout embryos, the dorsal foraminal cells fail to keep cluster, and they will migrate away from each other. So this explains why the top first vesicle formation has a defect in tetraspanian 4, tetraspanian 7 knockout embryos. So this data also rule out the possibility of DFCs problems because all of these DFCs are transplanted from Y-type embryos. So it's a cell non-autonomous manner. So if we summarize this data, we can conclude that migrasomes are required for establishing of left-right symmetry, and migrasomes are also required for the cup-first vesicle formation by regulating DFC's clustering. So we're wondering that which signal or say how migrasomes regulate the DFC's migration and the cup-first vesicles formation. We need to identify the signal enriched in migrasomes which can regulate the DFC's migration. So to identify the cargoes or enriched signals in migrasomes, we just purify the migrasomes from the Y-type embryo, and then we performed the quantitative mass spectrometry analysis to identify the enriched proteins in migrasomes. As we're showing here that in this volcano plot, we can see that the protein composition of the cells and the migrasomes are different. And in this red part, the red part represents the proteins which enriched in migrasomes. And we find that there are a lot of signaling molecules, including the chemokines, cytokines, growth factors, and morphogenes are enriched in migrasomes. So, which means that migrasomes enriched with signaling factors. This indicates that migrasomes may be enriched in some specified location and provide the regional signal center. Since I mentioned that migrasomes will accumulate in some specific location and to form some migrasome enriched center. So in this case, we focused on one particular chemokine, CXL12, because CXL12 is known to be very important in the early embryo development. So these beautiful images come from the cultured primary zebrafish cells stained with endogenous CXL12, which is highly enriched in migrasomes, as you can see that. And so, what's the exact function of migrasomal CXL12? To answer this question, we generate the CXL12 knockout fish, as well as the 6L4B knockout zebrafish. 6L4B is the receptor of 6L12. So we firstly checked the cup first vesicle formation. And we found that in the Y-type embryo, the cup first vesicle is normal, but in 6L4B or 6L12 knockout embryos, the cup first vesicle becomes smaller and the number of ciliaries become reduced and present cup first vesicle defect in this mutant fish. So which is this phenotype is very similar with the phenotype we observed in tetraspanian 4 and tetraspanian 7 knockout zebrafish. So we know that the migrasomal CXL12 is required for the formation of cup first vesicle. So we can summarize all of this result and think of a model that probably a lot of migrasomes enriched here and hold DFCs together. This model generates two hypotheses that the first one is we should see a cavity or a pocket enriched with migrasomes at the DFC site. And the second one is migrasomes should be able to hold the DFCs together. So we generate this hypothesis and we start to test this hypothesis by experiment. So for the first one, we just inject the high molecular weight dextran beads into the embryos to label the extracellular space within the embryo. And we can see that under the DFC's location, there is a big cavity is here. And if we label the migrasomes, we can see that a lot of migrasomes are enriched in this cavity. And to form a migrasome enriched center, just located under the DFCs. And we named this cavity with the embryonic shared cavity. So considering migrasomes are enriched in 6L12, this distribution potentially provides regional high concentration of chemokine signal to hold DFCs together. So we start to test the second hypothesis. To address this second hypothesis, we set up an in vivo chemotaxis assay. We just purify the migrasomes from the embryo and put into the agars just to make sure the migrasomes stay together and prevent the diffusion. And then we inject the migrasome contained agars into the ventral side of the embryo, where the DFCs and other cells shouldn't go during the gastrulation stage. And then we put the embryos for the 3D time-lapse imaging using light-shift microscopy. And we can see that in the control embryos, the agars which labeled with the dextran beads can't absorb the DFCs or other cells. While in the migrasome contained agars, the migrasomes can absorb the DFCs or other cells to the surroundings of the agars. And if we knock down the 6L12 signal in migrasomes, the absorption of the DFCs or other cells ability will reduce or disappear. So this result indicates that migrasomes enriched in the embryo cavity and hold DFCs and proper location to make sure the cup-first vesicle formation, and then contribute to the organogenesis. So which further support our hypothesis that migrasome enriched and then to form some signal in high concentration of signal to make sure that the DFC's migration and contribute to the cup-first vesicle formation. And actually, we have observed some similar phenotypes in the Tasmanian knockout mice. Yeah. So if we summarize all of this study, and we can get a migrasome working model that firstly, migrasomes formed during the gastrulation stage, and then migrasomes will detach from the retraction fibers to form some free-floating migrasomes. And these migrasomes will accumulate into some definite location to form some migrasome-enriched center. And then migrasomes will release the contained signaling molecules to form some regional cubes and regulate the surrounding cells, such as DFCs, and further contribute to the organogenesis. So why this is important? So if you remember that how the development biology works, it's just due to one single hypothesis, one key hypothesis, which is a signal diffusion model. And you can see that this fit map of the zebrafish. It's a very complicated map which is divided into different cell groups or cell fit. The diffusion model explains this with signal gradient formation between the dorsal to the ventral or as well as the anteriors to the posteriors. But it is still controversial and challenged by many people to interpret this complexity fit map. So if we think back to the migrasome, firstly, migrasome produced along with the embryo development. So migrasome possess the temporal information. And secondly, migrasomes can localize to some specific region cavity, which means migrasomes have the spatial information. And thirdly, migrasomes contain so many important signal factors, which means that migrasomes is an ideal information carrier, which can integrate temporal, spatial, and chemical information together and contribute to the embryo development. So we propose a new model based on the diffusion model, is that migrasomes can generate some regional cubes based on the diffusion gradient and form a more precise mechanism to regulate the cell fate. So if we summarize the zebrafish study, we can sort of what is migrasome. Migrasome is basically a delivery system. It puts the composition of factors to the right place in right time. So it is probably that migrasomes also involved in other physiological or pathological process, such as tumor metastasis immune response, because it's very similar logical that multi-types of cells migrate and need to coordinate each other. So again, we need to set up the research models and develop tools to explore the roles of migrasomes in other processes. So to explore more functions of migrasomes in different contexts, we established the intravital imaging systems and research models in mice. As you are showing here, that we set up the intravital imaging systems in mice, and we observed the migrasomes formation in living mice. We found that many types of immune cells actively migrate and produce migrasomes in the blood vessel. These migrasomes initially stick to the walls of blood vessels, but are eventually washed away by the bloodstream into the circulating systems. And these migrasomes may be essential for the many physiological process. And yeah, and the establishment of these animal models and imaging systems provide many support to migrasome studies. So if we come back to the intravital imaging systems, so in some cases, we can see that the problem which caused by the breathing and the heartbeat of the mice during the intravital imaging. So we need, as we're showing here, that you can see this problem. So we need an imaging system that possess higher temporal resolution, higher spatial resolution, and lower phototoxicity. We know that the lower phototoxicity is very important for living organisms imaging. And fortunately that the Dai group in Tsinghua University has developed a new scanning light field imaging system, which named with dot limit. The principle of this intravital imaging system is like the Drosophilus compound eyes. So there is a lens array consisting thousands of lenses is placed in front of the sensor, and which allowing and when the signals reflect from the samples and through the lens array, it will gather the signals to one focus, and then all of these signals can be accepted by the sensor. So this allowing real-time 3D volume imaging so that higher temporary resolution and lower phototoxicity can be achieved. And we also systematically verify the property of the new imaging system. As we're showing here, the dose limit shows that lower phototoxicity when compared to the traditional two-photon microscopy system and the traditional spinning disk confocal microscopy system. The dose limit imaging system can maintain very long time signals, as shown here. And in another case, we also set up an intravital imaging system using dose limit in zebrafish. We inject the tumor cells into zebrafish vessel and observed tumor cells generate retraction fibers and migrosome-like structures in the circulating systems. And in this case, we use the dose-limit system to perform intravital imaging in mice to observe the process of immune cells producing migrasomes. And we were able to obtain a higher quality time-lapse 3D image than the spinning disk microscopy system. We can capture the fine process of migrasome production. And in addition to this, a lot of interesting observations were captured by this imaging system, such as the circulating migrasomes, which may serve as a carrier for long-distance signal delivery in the bloodstream, and adhering of migrasomes to the wall of the blood vessels, which may provide local signals in physiological conditions. And another more exciting phenomenon is that the uptake of migrasomes by another cells. You can see that one cells produce migrasomes here, and then sometimes later, another cells will come to here and then internalize or engulf these migrasomes. So yes, the cells will engulf the migrasomes here. And we also observed that the division process of migrasomes. You can see here that one migrasome divided into two, so which may amplify the signals in physiological conditions. And then, what's more importantly, we injected the tumor cells into the mice and performed intravital imaging. We inject the tumor cells to mimic the tumor metastasis. And we can see that when tumor cells migrate in the blood vessel, they will generate migrasomes, and then these migrasomes will be internalized or engulfed by the following tumor cells. This observation suggests that maybe migrasomes play essential roles in tumor metastasis. And so all of these observations is under the study in our lab. And so the formation of migrasomes has been observed in various in vivo settings. And migrasomes have been shown to play important roles in cell-cell communication. And you know that we initially defined migrasomes as an organelle. So the functions of migrasomes extends beyond cell-cell communication. And I will give one case here that to show migrasomes play roles in maintaining cellular homeostasis. And it has recently been shown that damaged mitochondria can be selectively transported into migrasomes and expelled from cells undergoing mild mitochondrial stress. We named this fascinating process with mitocytosis. This process involves the relocation of impaired mitochondria to the cell's periphery and sequestered into migrasomes. And from this video, we can see that the process of mitocytosis, the damaged mitochondria under the mild mitochondrial stress, which caused by CCPR, relocated to the rear of the cell, and then the mitochondria were transported into migrasomes. So, considering the selectivity of damaged mitochondria to throughout from the cell, we raised a hypothesis that mitocytosis may be important for mitochondrial quality control. So, the answer is yes. We have identified that mitocytosis is very important for mitochondrial quality control during the mild mitochondrial stress. And to investigate the physiological roles of mitocytosis in vivo, we choose neutrophils as are subject to study, and they are highly migratory cells that are very abundant in the circulation. So to label the neutrophils in mice, we injected a fluorophore-conjugated antibody into the mice and used the spinning disk microscopy to observe the liver and the spleen. As we show here, we found that Our in vivo staining approach labeled the neutrophils efficiently, and the migrasomes were extensively generated by circulating neutrophils. And next, we tested whether neutrophils derived migrasomes contain mitochondria. We used mitosocs to label damaged mitochondria, and we found that many of the migrasomes generated by neutrophils contain mitosoc signal, which indicates that damaged mitochondria are transported into migrasomes. And we also purified migrasomes from the blood and performed Western blot analysis to check the mitocytosis. Accordingly, the isolated migrasomes contain various migrasomal and mitochondrial markers, as indicated here, which further supports the mitocytosis in physiological conditions. And we have identified tetraspanin 9 as a key regulatory gene in migrasome formation in neutrophils. So tetraspanin 9 knockout reduces the formation of migrasomes. So to investigate the physiological role of migrasomes, we generate the tetraspanin 9 knockout mice. And to directly compare the formation of migrasomes between wild-type and knockout neutrophils, we isolated the neutrophils from wild-type and tetraspanin 9 knockout mice and labeled these neutrophils with different colors. We labeled the wild-type neutrophils with the green colors, and we labeled the tetraspanin knockout neutrophils with the red colors. And then we inject the mix of the cells into the wild-type mice. We found that the formation of migrasomes by tetraspanin 9 knockout neutrophils is significantly reduced. And to test whether reduced mitocytosis affects mitochondrial quality control, we isolated neutrophils from bone marrow and spleen and performed the transmission electron microscopy analysis. We found that abnormal or damaged mitochondria are accumulated in spleen neutrophils in tetraspanin 9 knockout. But this doesn't apply to the bone marrow neutrophils. I will explain how this is happening. And we also measured the membrane potential by TMRM staining. We found the membrane potential is reduced in tetraspanin 9 knockout neutrophils, which isolated from spleen and blood. So to distinguish between the roles of tetraspanin 9 and migrasomes in mitochondrial quality control, we also measure the membrane potential in mature neutrophils in bone marrow. And neutrophils are produced and matured in bone marrow and then migrate into the circulation, where they undergo long-distance migration before reaching the spleen. We reasoned that if migrasomes, but not tetraspanin 9, are required to maintain mitochondrial quality, there should be less difference between the membrane potential of wild-type and tetraspanin 9 knockout bone marrow neutrophils, because bone marrow neutrophils have not yet undergone their long-distance migration, and therefore mitocytosis has not yet kicked in. So indeed, we observed that there is almost no difference in membrane potential between wild-type and tetraspanin 9 knockout bone marrow neutrophils. So this data suggests that it is migrasomes and not tetraspanin 9 that contribute to mitochondrial quality control. So finally, we explored the physiological consequence of impaired mitocytosis in neutrophils. It is well established that accumulation of damaged mitochondria can impair the viability of cells. Thus, we measured the number of mature neutrophils in bone marrow and the spleen of wild-type and tetraspanin 9 knockout mice. We found that the number of neutrophils in bone marrow is very similar in both genotypes, which indicates that the generation and maturation of neutrophils are normal in tetraspanin 9 knockout mice. However, compared to wild-type mice, the number of neutrophils in spleen is reduced, in tyrosine 9 knockout mice, which suggests that tyrosine 9 knockout neutrophils may have reduced variability. And the cell transfer experiment also presents a similar result, that tyrosine 9 knockout neutrophils have a reduced variability. This data suggests that mitocytosis does contribute to the variability of neutrophils in the circulation. So, in addition to the modulating cell-cell communication, migrasomes are also playing essential roles in maintaining cellular homeostasis by expelling unwanted substance to the outside of the cells. Yeah, of course, in this case, the migrasomes expelling the damaged mitochondria from the cells to maintain the mitochondrial homeostasis and to make sure the variability of the physiological conditions cell number. And another exciting discovery under study is the role of migrasomes in rare disease, such as hemophilia. And we know that hemophilia is a genetic disorder that impairs the body's ability to control blood clotting, leading to prolonged bleeding episodes. We know that hemophilia is a very serious disease that the people can't control the bleeding, and it's a very dangerous disease. And currently, the factor VIII gene knockout mice serve as a crucial mouse model for studying hemophilia. And to investigate the role of migrasomes, open the potential therapeutic application in this hemophilia, we get this factor VIII knockout mice from GemPharmatech and investigate the roles of migrasomes in hemophilia. And to test the roles of migrasomes in hemophilia, we established the bleeding assay as we're showing here. That in this model, we surgically incise the femoral artery of mice to observe the impact of migrasomes on hemostasis. Post-incision, there was an immediate ingress of blood, and after 1 minute interval, we cleared the hemorrhage using absorbent paper and promptly applied migrasomes to the wound site. And remarkably, hemostasis was achieved within 100 seconds and post-migrasomes treatment. In contrast, the control mice presented persistent bleeding. So, which means that in control mice, it's a factor VIII deficient mice. The mice will keep bleeding after incision. But in the migrasome treated mice, the bleeding will be prevented within 100 seconds. So this observation not only suggests that it's the essential role of migrasomes in coagulation, but also underscores their prospective utility in hemostatic intervention. So it could be possible to develop migrasome-based hemostatic drugs. So this research is still ongoing, and I look forward to further communication with you in the future. So overall, we can get a summary that what migrasome can do. I think that migrasome is an information vehicle which can modulate cell-cell communication by providing local signal and maintaining cellular homeostasis by expelling unwanted substance from the cells. The informations or cargoes or signals which contained in migrasomes define the roles of migrasomes in various biological processes. Yeah, actually, we have many ongoing projects to study the roles of migrasomes in different physiological or pathological conditions. And eventually, and especially, we also started to investigation of migrasomes roles in human disease. And I hope we can get some interesting results or have a very promise progress in this study and to contribute to the human health and the human disease therapy. So in the end, I would like to thank many people. That's Professor Li Yu and our great collaborators and all of the lab members. Yeah. Thanks.
Okay, thank you, Dongjiang. And any question from our audience, please, if you have any questions, just text them in the text box.
Okay, any questions?
Coming. So if I can start some of the part of the conversation now by discussing something that I'm particularly interested in. I don't know if it's okay. So if there's any questions from our audiences, so then if they have this in the chat box, they can interrupt us in any time during our discussion. But now, so very elegant work. But from this elegant work, so the first question from me is that so because some sort, I think that the migrosome itself is somehow a little bit like the exosome itself instead, but it is linked to the cell which secreted with the fiber, and also the size of this organelle is comparatively larger compared to the exosome itself. My question is, so the first basic one is, how do you purify them? So we know that there's some exclusive markers for exosomes like CD63 or something else, but I don't know if there's some, you mentioned some markers like the spanning proteins of the migrosome, but I don't know if they are exclusively expressed in these organelles and if they can help you to purify them or not. So this is one of the basic information I want to know from your study.
Yeah. It's a very good point that it's during when we discover the migrasomes, the first question is we ask, what's the difference between migrasomes and other extracellular vesicles, of course, including the exosomes. So we published the paper to identify and to describe the difference between migrasomes and the exosomes. So in summary, that the difference is the first one is the morphological difference. The exosomes should be 100 nanometers, right? But the migrasomes one is 1 micrometer. It's very different. The migrasome is bigger than exosomes. And the second one is the biogenesis process. Migrasomes are generated from the retraction fibers, and which is connected to a fiber. So it's the most typical morphology characteristic of migrasomes, which is a vesicle connected to a fiber. So the exosome is just a vesicle, right? And the third one is the regulated gene is different. When we knock down the migrasome regulated genes, such as the escort complex, there is no effect on migrasome formation. So of course, when we knock down or knock out the migrasome genes, it doesn't affect the formation of exosomes. Yeah, and so we know the migrasomes and the exosome is very different. So, and you also mentioned that how we purify the migrasomes from the samples. As when we purify migrasomes from the tissue culture systems, we discard the supernatant of the cell culture medium. So the exosome purification is from the supernatant of the cell culture medium. So it's different, right? And for the second one, when we purify the migrasomes, we use the 20 RCF centrifugation, which is not sufficient to spin down the exosomes. So the exosome fraction and the migrasome fraction is totally different. Yeah.
Yeah, okay, I see. So
I
just like
see.
gradient centrifuge, we separate different compounds with different molecular weights or different size by
Yeah.
a gradient centrifuge. Okay,
Yes,
I
Yeah.
see.
So you mean
But now still we
you first?
please.
The physical character of the migrosome and the other vesicles are different, so you can isolate them from these characters, right?
Yeah.
Okay. Intro?
Okay, so that's very good. So I understand this. So the second question is that so I used to work with zebrafish a lot and also for development biology. So I think that this addressed a very good question is that because traditionally, we know that all those, like chemokines, but especially for the morphogens, they works, in the development itself, like the gastrulation part and forming of, the body segments, and then eventually the organogenesis process, the different, morphogens and morphogen gradients, they together, form the pattern of the early development. But now this, gives us a new hint that the, morphogen gradient, the restriction of this morphogen gradient may not, due to some, biochemical process, but rather, a physical process by the migrosome to, restrict it to a, certain physical, part or physical, region to, restrict this gradient. But my question is that we know that the majority of the morphogens, including Wnt, including the BMPs, TGF betas, they work as membrane proteins and they need to bind to the receptor of the cells. So, but it shouldn't be functional if they are kept into the, cargo of the migrosome. So my question is, do you know, are you focusing on how the migrosomes release these kinds of functional molecules to, the biological fluids like the blood or the,
Yeah, it's a very fundamental questions in migrasome biology. Actually, we have a paper. It's in the revision stage in the cell. So we know that the cargos or signal factors, just like you mentioned, the BMPs, Wnt, and the TGF-betas, they contain in migrasomes. And when the migrasomes get into the location, they function, they will release the signaling factors to the extracellular environment. Yeah, and there are two ways of migrasomes to release the signaling factors. The first one is the migrasomes will rupture and to expose all of the contents. And the second one is we find migrasomes membrane is leaky. So the small molecular weight proteins can leak from the migrosome membrane and get to the out cellular space and can bind into the receptors. So why these signaling molecules can contained in migrasomes can be functions. Yeah.
wow, that's amazing. So I actually can't wait for your paper to be published, so I definitely read it.
Yeah.
There's
This, yeah.
there's a
So,
question.
but no, yes, from our guest
Yeah. Can you see that? Could
in the chat
mitocytosis
box.
happen also for uh, don't could you see that the question from our audience?
No, I can see that. Oh yeah. I can see that.
Yes, I think
Yeah.
so. The,
So
the morphology
uh
of the cell itself.
actually for these questions that the he asked that could mitosis happen also for other cells like microglia? Do you think that microglia which have many protrusions could form migrasomes? Yeah, actually we have projects to study the migrasome formation in microglia. And so far we don't know mitocytosis. Does mitocytosis happen in microglia? And we don't know the protrusions of the microglia and the difference between the migrasomes retraction fibers and the protrusions. So actually we don't know. But if I can make a speculation that just maybe, the microglia also possess mitocytosis process to maintain the cellular homeostasis. and but it's just a speculation and no data to support. Yeah.
Thank you. So, okay, based on your now the discussion, so I also have two questions, one fundamental but one experimental. So for the first one, let's give the experimental one. So because we have seen that, or you have seen the the morphogens and the chemokines to be observed in the proteasome of the microsome. But what I want to know is that if they are enriched, so compared to the cytoplasm itself from its origination, so if there's any enrichment of these proteins into this like an extracellular organelle. So that's something I'm wondering. So if it is the case, maybe we can use this as a therapeutic vector or something like what exosome is doing, but
Yeah.
with a relatively
Yeah.
larger content,
Yeah,
larger
yeah, yeah.
load compared to the exosome.
Actually, we have identified the mechanism to sorting the migrasome cargoes, and also have engineered migrasome pattern. Yeah. So, and another lab members are trying to try the delivery systems, deliver the cargoes or drugs or anything else to by migrasomes. Yeah, it's maybe.
Could
Okay,
you
so
reconstitute this the migrosome in vitro?
Yes, we can reconstitute the migrasome image. We the Huang Yu paper published the NCB paper in 2019 that reconstitutes the migrasomes in vitro using the lipids and mixed with the tissue spinning for proteins. Yeah.
Okay.
yeah. So another question from our audience is that, you know, he or she wants to know that the leakiness of the membrane of the microsome, does it happens passively or regulated? So
Yeah,
do you have
it's
anything?
a very it's a very good question. We don't know. Yeah.
Yeah,
In so far, we want to know whether this process is regulated by the cells,
yeah, sure.
but so far we have no data to show whether this process is passively or
Yeah.
actively.
We
So we
can,
don't know.
yeah, we can see that, yes, because this process itself is novel enough, and this field is relatively
Yeah,
not that
yeah,
popular these days. So maybe we need more people to focus on this field. So observing this
yeah,
phenomenon itself is, advanced enough, I suppose. So maybe, the scientific field they need to, pay more attention to this
yeah. I
special.
think this process needs to be regulated because if this is
Yeah,
passively and no regulation, it will be it will bring some troubles in
yeah,
many
maybe sometimes,
regions, right?
yeah,
Cell
a
body
severe disaster during early embryogenesis. I can,
Yeah,
tell this for sure.
Yeah,
Okay.
I agree with that. I think all the process in the cell are under control because if there is no this regulation process, many metabolic pathways will be out of control to cause some cellular damage, I think.
yeah. I
Just
agree with this. Yeah,
yes, the difference is that some are autosomal, but some of them are passive or responsible, responsive. No, I'm sorry, responsive. It's just passive things,
yeah.
or aggressive things. I don't know. So then there comes my last fundamental question. So because its influences are sometimes cell migration, or sometimes regarding to the asymmetric development of the body axis itself. So may I suppose that also the the generation or origination of a migrosome is also asymmetric, from its parental cell. But so this asymmetric in the parental cell's membrane. So do you have any idea how it may happen?
Uh, Sorry, you mean the how the parental
Yes,
cells produce
the
microsome?
origination of the the migrosomes are asymmetrical from the parental cell, right? I
Parental
suppose
cell.
from, yes, from the originating cell, the cells who have this, who secretes these microsomes. the secretion or generation of the microsome may be asymmetrical from your video and your data, and also the theoretical consequence of these microsomes. So how did the cell knows where to grow or where to form these microsomes?
Yeah,
In
yeah,
which
yeah.
direction or
Yeah, it's a very good point. Actually, I didn't mention in this talk that migrasome formation is depends on cell migration, right? And another point is migrasome formation is needed to adhesion to the extracellular matrix. So
oh,
in the living organisms, especially in different tissue or different organ, the extracellular matrix distribution is different. So this difference of the extracellular matrix distribution maybe can determine the formation site of migrasomes. So when the cells migrate through the different site of and distributed with different extracellular vesicle, can produce different migrasomes. Yeah.
wow, yes, amazing. So maybe they are interacting with some catecholamines or something else. So the
Yes.
fiber
Yeah.
or the membrane of the microsome. Okay, I see.
Okay, so it is a special day today, rare disease day. So, what's your next plan? Do more research based on microsome function in multiple diseases, including rare diseases?
Yeah. So I'm sorry. So you mean the migrasomes studying different diseases?
Yes.
Yeah.
So actually, we have many studies of the migrasomes in some relevant disease. Just like I mentioned, that's hemophilia, and in addition to the hemophilia, also are ongoing the investigation of migrasomes in some related disease, just like the tumor metastasis, tumor immunity, and some other coagulation related disease. Yeah. So I think maybe when the studies get some promising progress, we can share with you in the near future. Yeah.
We are looking forward to that. How
Yeah.
about
So
you?
you did, yeah, sorry.
Uh,
So your
w-
data,
w-
regarding the, hemostasis is very interesting. So because as our traditional information about how the blood coagulates after damage, so there's a positive feedback about all the factors they enlarge each other, and then accumulated effect, how the blood coagulation functions. So I wonder, so maybe the migrosome, they supplied one or multiple of the terminal functional molecules during this positive feedback, and then itself mediates the coagulation of the blood. But if there, I don't know if there's any other possible mechanism, since the model you used, they are deficient in factor 8. So other than supplement of these downstream signals, I cannot find any way currently to explain this phenomenon. So I don't know if you have other ideas, like
Yeah. Regarding to the coagulation, actually, we have a systematically study of migrasomes in coagulation, and this study is the paper is in revision in the Nature, and but we and in summary I can say that migrasomes is a very essential component of the coagulation systems, and migrasomes enriched with a lot of coagulation factors, so
wow.
it can play essential roles in the coagulation site. Yeah.
Oh, that's very amazing. Not regardless of the origin, how you make them. So it is very maybe maybe can become a universal cure for hemophilia regardless of the type, the mutated gene factor 7, 8, 9, 11,
Yeah,
regardless
yeah,
of this kind of factors. This is very good,
yeah.
amazing information.
so, any question from our audience? Okay, let's just finish today's webinar. Thank you, Dong, and thank you, Bingzhou.
Thank you. Thank you all for attending our webinar. And thank that you focus on GemPharmatech and wish that we have a further collaboration with all your audience.
Thank you. Have a good day, everyone. Bye-bye.
Yeah, thank you. Bye.